Our bodies are incredibly complex machines, built and maintained by intricate instructions. These instructions are encoded within our DNA, organized into units called genes. Genes are the fundamental blueprints that dictate everything from our eye color to the proteins that keep our hearts beating. Sometimes, a tiny error in just one of these crucial blueprints can have profound effects. This is the essence of a single gene disorder. These conditions arise when a mutation, or change, in a single specific gene leads to a particular disease. Imagine a gene as a recipe for a specific protein. Proteins are the workhorses of our cells, performing countless functions. They build structures, transport molecules, catalyze reactions, and send signals. Each gene contains the precise sequence of DNA bases that tells a cell how to assemble a particular protein. When this sequence is correct, the protein functions as intended. This intricate system ensures our bodies operate smoothly. A mutation is a permanent alteration in the DNA sequence that makes up a gene. These changes can be as small as a single base pair substitution or as large as the deletion or duplication of entire segments of DNA. Such alterations can occur spontaneously or be inherited. When a gene's blueprint is altered, the resulting protein may be malformed, non-functional, or even absent. This disruption in a single protein's function can cascade, leading to a specific set of symptoms and ultimately, a disease. Single gene disorders are categorized by their inheritance patterns. One common type is autosomal dominant inheritance. In these conditions, only one copy of the altered gene is needed to cause the disorder. If a parent has an autosomal dominant disorder, there is a 50% chance that each child will inherit the altered gene and develop the condition. Examples include Huntington's disease, which affects nerve cells in the brain, and Marfan syndrome, impacting connective tissue. Another significant category is autosomal recessive inheritance. For these disorders, an individual must inherit 2 copies of the altered gene, one from each parent, to develop the condition. People with only one copy are called carriers. Carriers typically do not show symptoms but can pass the altered gene to their children. Cystic fibrosis, affecting mucus and sweat glands, and sickle cell anemia, which impacts red blood cells, are well-known examples of autosomal recessive disorders. X-linked disorders are caused by mutations in genes located on the X chromosome. Since males have one X and one Y chromosome, and females have 2 X chromosomes, these disorders often affect males more severely. Females with one altered X chromosome are usually carriers and may have milder symptoms or none at all. Hemophilia, a bleeding disorder, and Duchenne muscular dystrophy, causing progressive muscle weakness, are classic examples of X-linked conditions. The specific symptoms of a single gene disorder depend entirely on which gene is affected and what protein it normally produces. For instance, a faulty gene involved in blood clotting leads to hemophilia. A gene mutation affecting lung function can cause cystic fibrosis. These disorders can impact virtually any body system, from the nervous system and muscles to metabolism and organ function. The severity and progression of symptoms can vary widely, even among individuals with the same condition. Diagnosing single gene disorders often begins with a thorough review of family medical history and a physical examination. Genetic testing plays a crucial role in confirming a diagnosis. This involves analyzing a person's DNA to identify specific gene mutations. Genetic testing can be performed using blood, saliva, or tissue samples. It allows for precise identification of the altered gene, which is vital for accurate diagnosis, prognosis, and treatment planning. While many single gene disorders currently have no cure, significant advancements have been made in managing symptoms and improving quality of life. Treatments often focus on alleviating symptoms, preventing complications, and supporting affected body systems. For example, individuals with cystic fibrosis receive therapies to clear airways and improve digestion. Those with sickle cell anemia may receive pain management and blood transfusions. Research into gene therapy and gene editing holds promise for future curative treatments. Genetic counseling is an invaluable resource for individuals and families affected by single gene disorders. Genetic counselors provide information about inheritance patterns, risk assessment, and available testing options. They also offer emotional support and help families understand the implications of a genetic diagnosis. This guidance empowers individuals to make informed decisions about their health and family planning. Understanding single gene disorders is a cornerstone of modern medicine. As our knowledge of the human genome expands, so does our ability to diagnose, manage, and potentially cure these conditions. The ongoing research into gene editing technologies like CRISPR offers exciting possibilities for correcting the very genetic errors that cause these diseases. The journey from one gene, one disease, to one gene, one cure is a testament to scientific progress and hope.